You're listening to the Effective Statistician podcast, a weekly podcast with Alexander Schacht, Benjamin Pieske, and Sam Gardner, designed to help you reach your potential, lead great science, and serve patients without becoming overwhelmed by work. Today, we are talking about the analysis of adverse events done right. Really interesting interview with Caspar and Jan, so stay tuned. When I started in my career as a statistician in the clinical world, I was wondering about safety analysis. And then I digged a little bit deeper into it, and then I thought, well, that's always the same. We just count the patients who have an event, job done, and then just repeat. But it's not that simple, especially in more complex situations where you have different follow-up times and things like that. Then the patients that actually stay longer on treatment and that helps them get more adverse events, and it looks like the adverse event profile is worse for the really good treatment. So how can you account for that? Stay tuned for this really nice interview with Caspar and Jan, who are both experts in this area. You can also check the show notes because there's lots of additional links and resources that we are mentioning throughout this episode. I'm producing this podcast in association with PSI, a community dedicated to leading and promoting the use of statistics within the healthcare industry for the benefit of patients. Join PSI today to further develop your statistical capabilities with access to the video-on-demand content library, free registration to all PSI webinars, and much more. Head over to the PSI website at psiweb.org to learn more about PSI activities and become a PSI member today. Welcome to another podcast episode of The Effective Statistician. Today we are talking about a very interesting topic, and for that we have a couple of very interesting guests. But first, hi Benjamin, how are you doing today?
Hi Alexander, very well. It's a nice sunny morning. It's really like spring is finally coming, I hope. I mean, it's in the middle of April when we record this episode, but it's quite nice today.
Yep. And we have Jan and Caspar here. Caspar, you have been on the podcast already once, so great to have you back.
Yeah, hi everyone, and thanks for the invite.
And Jan, someone from Ulm, quite close to where I lived for 2 years in Biberach. Welcome to have you also on the show.
Yeah, hi everyone. Thanks for inviting me too. And it's my first time.
Yeah, great. Jan, maybe you can speak a little bit about your career up to now and what got you involved into this project.
Oh, so I'm professor of biostatistics at Ulm University, where we have a study program, mathematical biometry. So we have a substantial interest in biostatistics and applications in the life sciences, including that in the pharmaceutical industry. I'm originally a mathematician by training, started out in mathematical statistics. That was something between black and gray, so I turned to more applied stuff and obtained my PhD in Freiburg while at the medical biometry unit there of the University Hospital Center Freiburg. And let's say I'm interested in all things event history analysis. So that's a very brief sketch.
Okay. Yeah, then that fits perfectly into the topic of course. And Caspar, you have been on the podcast already, so you can scroll back a little bit to learn more about him. And yeah, today we are talking about something that it's a really interesting thing. It evolved over a long period of time and was a hot topic for quite some time. It has something to do with estimates, and this has something to do with oncology. And the acronym we are talking about today is, and I'm not sure whether I say it correctly, SAVI. Or how do you pronounce it?
Caspar, SAVI, yeah.
SAVI is correct.
I mean,
That's the term we've been using
in German Swiss English. But I think we're all not native speakers, but yeah, savvy is correct, isn't it? Or savvy.
Well, what does it stand for? Maybe the question from that side.
Yeah, survival analysis for adverse events with varying follow-up times.
And you'll find the letters in that order in this long sentence if you are creative.
Yeah.
Okay, so varying follow-up times. So that means you have basically a study where you're comparing two different treatments. Say, two different groups of patients get into two different groups of treatments. And then for whatever reasons, one treatment has, let's say, a median duration of 6 months, and the other one has a median duration of 12 months. And so if you look into kind of time to end of treatment, these Kaplan-Meier curves look very, very different to each other. So how is that a problem in oncology? And where is this problem coming from in oncology? Caspar, maybe you want to start?
So one way to look at this very simply is, imagine you have a treatment that is very effective. So you have an overall survival hazard ratio of 0.5. So on average, if we make things very simple, every patient on the treatment arm is under observation for double the amount of time compared to a patient on the control arm. Now, if you look into the occurrence of adverse events and you simply count the adverse events and divide them by the number of patients, a quantity that we call incidence proportion and that we are all very familiar of using in clinical trials, of course, you most likely will find more adverse events in that group that has double the survival. So ultimately, the question is that a fair comparison? So that is kind of a key question. If the follow-up is so different or the time at risk is so different, just counting the number of patients with a given AE divided by the group size might not be estimating the thing we want to potentially know, namely the probability of an adverse event in an appropriate way. But before I hand over to Jan, I would want to make one other comment. This is not at all specific to oncology. This applies to all kinds of therapeutic areas where we are interested in assessing the risk of an AE.
Yeah, okay. Or maybe not even an AE, could be anything, isn't it? Any event.
If you're interested in the probability of a certain event happening, maybe we can discuss about that also later.
Yeah, I fully agree with that. And I think one starting point for Savi was that although essentially evolving in the very same trial, we're using different statistical methodology for efficacy and for safety. That is a puzzling thing, to say the least. But to back up what Caspar has just said, Savi evolved from a couple of different projects or workshops maybe, and one was a workshop of the working group therapeutic research of the German Society for medical informatics, biometrics and epidemiology, GMDS, and they had a workshop on this topic in, I think, 2014, and that evolved in a special issue on the analysis of adverse events in pharmaceutical research. And I'm talking about that because Ralf Bender, Lars Beckmann and Stefan Lange from the IQWiC, the German HTA, they have a very interesting paper in that special issue, and it describes examples that Caspar has just explained. There's one example where arguably the increased proportion of adverse events in the experimental was due to the fact that the treatment was effective. There was substantial prolonged survival. And for me, the freaky thing, as explained in that paper, was that the company to some extent discussed that the safety profile of their drug was worse. But the reason really was prolonged survival in the first place. And on the other hand, the IQWiC, the German HTA, was not too happy with the analysis, but to the best of my knowledge, they never solved the issue. And this is where Sevi wants to make a contribution, because it is almost absurd, you could say, that you have safety concerns about a drug, but the safety concerns maybe solely stem from the fact that you have prolonged survival, and if these are initially truly ill patients, as cancer patients are or may be, then you will also see more adverse events. But as Kasper said, I want to back that up too. I guess oncology may be a major field, but I'd say whenever you have a treatment effect that prolongs survival or prolongs time to event in general, that might also have an impact on the monitoring of adverse events, and where you have what we call competing events. Could, for instance, be death from other causes if the primary outcome is cardiovascular death, or let's say in trials in COVID-19 treatment, the aim is to speed up recovery. So you have a time to event. If that duration is, in this case, not prolonged but shortened, that is much better for the patient. But patients may, of course, also die. And then you have all these things together that might compromise the analysis of adverse events if you're using overly simplistic methods.
So it could also work in the other way around. Yeah. So if you have, whenever you respond, you stop treatment, and then you have the time to response, and you measure only the adverse events under treatment, then of course a very effective treatment would have much less exposure time than not so effective treatment. And so you would have then less adverse events on the more effective treatment, just because you have less exposure, isn't it?
I would think so, yes. I mean, typically we think the other way around. We think survival, we think time to death, and then prolonging survival is the thing to go for. But yes.
Yeah, and maybe I can add to that. Very interestingly, when I train people internally on this topic, I would often ask, if we want to assess whether a treatment prolongs survival, would we just kind of have a randomized trial, staggered entry, and then we cut the data after 3 years, and we would just count those who died, divide by the group size, and then compare these two proportions? And everybody would say, that's not how we should do things. We would never do that. But if you then move on to safety, that's exactly what we do. So that hopefully illustrates this discrepancy between efficacy and safety. And that also surfaces very, very quickly. Sometimes we have trials where an AE type of endpoint is the primary endpoint. And then very quickly, the discussion is very different from this usual AE analysis we are doing. And it's just striking that we are using kind of one type of mindset for efficacy, and for safety, we don't seem to apply the same kind of mindset. And this is even more striking, as the methods that we should use to account for all these features, kind of varying follow-up, staggered entry, competing events, as Jan was saying, these methods, they are established for decades. That's maybe also something we can talk on later. So it's very, very surprising that, at least to me, that we are not given the enough priority to how we analyze safety data. And even more ironically, that one method that we often use to estimate the probability of event actually overestimates that probability of event. So that's this famous 1 minus Kaplan-Meier estimate. So we are kind of punishing ourselves for no good reason, and that is surprising to me.
I find it really interesting what you say in that regard, that I was always thinking kind of from the estimand situation. In the end, you are interested in a benefit risk assessment. And if you have a benefit risk assessment, I think you have the same kind of estimand approach for the benefit as for the risk, because you want to have the same kind of decision in mind. You want to have the same population in mind. Of course, you have different endpoints in mind, but kind of you want to have kind of the same strategy approach there. because otherwise, what's the decisions that you are actually evaluating? What is the kind of, you can't have for the benefit one decision and for the risk another. You can only have one decision. Yeah, you can't choose whether if you select your treatment, you can't choose for the efficacy I'll want to have that thing, and for safety you want to have the other thing. You can only have one. Yeah, so I think that is really important, that we have a more unified approach in terms of the estimates on both the efficacy and the safety side.
Very good. What is then the kind of the starting point in your working group to really handle the problems that we just mentioned, or maybe combine the different views of the methodologies, but also looking one side efficacy and the other side safety at the same time? So what is the approach that you are taking there then?
I completely agree to what you're saying, Alexander. And I think one aspect that we need to keep in mind is, for efficacy, we focus so much on determining a primary endpoint and maybe a few key secondary endpoints. And then we invest a lot of energy in properly define these, properly assess these. And I think one aspect that needs to be discussed in this context is, what is actually the goal of our safety analysis? And I see at least two goals. One goal is kind of rough signal detection, if you want. And I think when we think of how we routinely report safety in clinical trials, we have these endless tables where we have all these AEs, and then we look at the proportion, this incidence proportion in the two arms, and maybe some kind of relative effect measure. And then we try to filter those out that are maybe different. And then we start to assess, is this difference now clinically meaningful? You have all these statistical challenges with this approach, like I wouldn't call it multiple testing, but if you look at many things, just by chance, some of them will pop up, even if there is no underlying difference. So that is one goal of safety. The other goal of safety, and that is maybe what would more relate to a benefit risk assessment, is if you define a small set of AEs of interest, for which you then want to have an accurate assessment or an accurate estimate of the probability of that AE actually happening. And then you would, I think it will make sense to invest more energy there as well, and define these endpoints properly, define the estimate, define the thing you want to estimate. And that is one of the big benefits of the estimand addendum, then align the data collection strategy to the thing you want to estimate. And my call here would be that we make a distinction between these two goals, and not just look at these tables of AEs. And then if you are actually interested in a probability of AE, just take the numbers from these tables. I think then we would need to account for competing events, staggered follow-up, and all these aspects, and properly estimate the probability of an AE.
I mean, is that a methodological question? So what do we do in terms of statistics, or are you interested in the kind of fun discussions that we have? Pardon?
Let's start with the methodology. No, let's start with the methodology, kind of where this is aiming at.
Right. Let's see. I think for me, one basic step is to really remind ourselves why we do survival analysis in the first place. And Caspar had a nice argument in a nutshell early on when he explained how, when he does in-house training, that everybody agrees that for time to death, you should not simply count the number of deaths and divide by sample size, because we have censored data. And that really is the starting point. So, I mean, the usual textbook tale is we have time to death data. Alas, for the statistician, not everyone dies. So we have an information loss leading to censoring, and then we do something else. And then that often in the textbooks is Kaplan-Meier. But the starting point here is that this is true for both efficacy and safety for the occurrence of adverse events that is connected to varying follow-up times, and the methodological backbone of survival methods and also of Kaplan-Meier is the analysis of hazards. So the way we go about this is looking into the hazards of adverse events and let's say any event that may preclude or stop observing adverse events, let's say such as death, before you have experienced the adverse event that you're currently investigating. And put simply, one method I understand in safety analysis is to look at the incidence density or incidence rate, as they call it. So number of adverse events, let's say first adverse events divided by patient time at risk. And that is a very simple parametric hazard estimator, but you could also do it, let's say if death is that other event that may happen before you have that adverse event, you also do the calculation for the death event. And then you have two incidence densities and you try to put them into perspective. And going back to that paper by Ralph Bender that I talked about, where they had prolonged survival and an increased number of adverse events, you might have the same incidence density of adverse events, but if you have a prolonged survival and that incidence density of an adverse event is unchanged, it ticks at the same rate, if you will, and if it does so every day anew and you have more days, you will see more adverse events. So that disentangles matters.
So it really depends also on when these events occur. Yeah. So I think if you would have a typical event that only occurs, directly after start of treatment, it actually wouldn't change a lot. Yeah, because you don't have any censoring usually, or a lot of censoring very, very early in treatment. So then the analysis would be the same. But if you have events that only occur quite after some time, then the effect becomes even bigger. Yeah, because then you have much more people censored before the events even have the opportunity to show up. And so if there's a certain method of actions behind where kind of more the cumulative dose over time triggers something, then these types of adverse events, you have a completely distorted view of things.
Yes, there are a number of factors coming into play, and as you say, if everything occurs into a well-defined time span, and if that is not subject to censoring, if these time spans are essentially identical, at least across arms, if not across patients, preferably so, then I'd also say that a lot of these difficulties disappear. However, how these possible sources of possible bias actually affect your results, that is a quite involved thing to investigate. And so what we've done in Savi is come up with a methodological meta-analysis across a lot of pharmaceutical companies, and where we have tried to, or where we aim to investigate these differences in investigating AE risk using different methods. And we also investigated the possible impact of, the amount of censoring, the amount of what I call a competing event, such as death, the impact of follow-up time, and Kaspar, maybe you can shed more light than I can, but it is a pretty involved picture. One reason being that, of course, the amount of competing events also depends on the amount of censoring and vice versa. And let's say A colleague of mine, Thomas Gertz, when asked about using more sophisticated methods, his answer is if you have fake money in one pocket and real money in the other pocket, which money do you use to pay what you're currently buying? And he prefers the real money. So maybe, I guess there are situations where I shouldn't say overshoot, but maybe the methods we're talking about here are not always needed, but they will typically not go wrong.
Okay. Yeah, I think that's a great recommendation. Make it as sophisticated as needed, not as possible.
Yeah.
Caspar, so maybe you can speak a little bit about the different approaches you have looked into. Mm-hmm.
Yeah, and exactly. So maybe it's time to also give some indication about how large are these biases. If we say we should make it as simple as possible, and does it actually matter? Do we need more complicated methods, if you want, although those methods that account for all these features are not that complicated? So the way we approach this in Savvy is that 10 sponsors, 9 pharmaceutical companies, and 1 academic trial center provided 17 randomized trials that have finished, read out already, and from all therapeutic areas. So it's not only oncology, it's also other therapeutic areas like cardiovascular and I think MS, multiple sclerosis, etc. And then we defined a few AEs in all of these trials, and we defined a set of competing events. And then we applied a bunch of estimators to these AEs and benchmarked them against what we call the gold standard. And the gold standard is the Allen-Johansen estimator, because that is a non-parametric estimator that accounts for varying follow-up time, censoring, and competing events. And you can think of the Allen-Johansen estimator as a straightforward non-parametric generalization of Kaplan-Meier to the situation where you actually have competing events. Okay, so if we first start with the incidence proportion, that is the prevalent estimator we use, or estimator. It's actually not really an estimator, it's just the prevalent approach we use, because we are not so clear what we're actually estimating in safety analysis very often. But that is this number we compute by just counting the number of AEs divided by the group size. And if you benchmark that against Allen-Johansen in these 17 trials, and we actually looked at 186 AEs combined from these 17 trials, you find that the incidence proportion generally underestimates the probability of an AE, and that can happen up to a factor of 3. So instead of you have a 12% probability of an AE that you would receive with Allen-Johansen for a given AE, sometimes when you just use this incidence proportion, you only get 4%, okay? So that gives you an indication of the extent of the bias for the incidence proportion. If on the other hand, you estimate the probability of an AE by just taking Kaplan-Meier and censoring at competing events. So if a patient dies, you would just censor that patient and then you compute time to AE. We know this is not what you should do. This has been discussed in the literature extensively. But what we find in Savvy is that with this 1 minus Kaplan-Meier estimate, you actually overestimate the probability of an AE up to a factor of 5 sometimes. So why would you want to do that? Just because you're lazy, you're just using a method you know very well, you censor the competing events, sometimes you overestimate by a factor of 5. And this is even worse, as the kind of unbiased estimate is pretty simple. It's available in all software packages, and you need to invest a little bit. You need to properly define the competing events, but then you can actually use that. And the follow-up question then is, so this is estimation of an AE probability in one arm, in one group. underestimation using the incidence proportion, overestimation using 1 minus Kaplan-Meier. But what happens then if you divide the two when you want a relative comparison between two treatment arms? And there, it's interesting that for the incidence proportion, on average, you actually do quite well. So you divide two biased estimators, and then you, very loosely speaking, you end up with a halfway unbiased estimator. But that wouldn't justify the use of the incidence proportion still. But this is just what we found in SAVI. Okay, and for Kaplan-Meier, typically you underestimate the relative effect. And eventually what we then did is eventually ends up in labels, in drug labels, are kind of labels like an adverse event is happening very rarely, uncommonly, commonly, or very commonly. For example, these are SMPC frequency categories put forward by EMA. And you then categorize these 186 AEs into these categories using the incidence proportion and using Allen-Johansen. And then you find that sometimes you really would make a different decision. Okay, so that doesn't happen so often for estimation of an AE in just one arm. And these tables, you find them in the SAVI publications, but it happens actually to a quite scary extent when you look at the relative measure. So I can just read off that table. If you take the gold standard Allen-Johansen estimator, you estimate the relative risk, sometimes you conclude that this risk is major from the treatment compared to the control. And if you would use the hazard ratio from a Cox regression, where you just censor the competing events, you would actually estimate in certain cases that this risk poses no difference. So you either had a major difference using an unbiased method, and with the kind of standard method that we often use, you would conclude there is no effect between the two arms. And in my opinion, that is quite scary and something we really need to look into.
Can you, for those that are not so familiar with time-to-event analysis, can you explain a little bit what the Allen-Johansen estimator does differently to the Kaplan-Meier estimator, which probably all are familiar with?
In a nutshell, what Kaplan-Meier aims to do is, or 1 minus Kaplan-Meier aims to approximate an empirical distribution function, number of events up to a certain point in time divided by sample size. And the approximation comes from the fact that we do not know the number of events. We only know the number of observed events. And the reason is censoring. So 1 minus Kaplan-Meier is approximating this.
Yeah, in a nutshell.
And now, if you look at the step functions of Kaplan-Meier, they are, of course, you have these steps when you observe events. And all you do is this approximation of an empirical distribution function. You split that up in additive fashion in your approximation of the empirical subdistribution function for, let's say, an adverse event, plus the empirical subdistribution function for, let's say, death before the adverse event could have potentially occurred for that patient. And I think a good check always is to have a look into what happens if you have no censoring at all. And then what Allen-Johansen does is, it is simply the number of adverse events up to a certain point in time divided by sample size, which is perfectly okay in the absence of censoring. And Kaplan-Meier is the number of survivors divided by sample size. And that eventually, over course of time, if you track down everyone, will drop down to 0, because everyone dies at the end of the day in a super long trial, let's say, but not everyone would experience the adverse event under investigation. That is the reason for the bias that you have when using 1 minus Kaplan-Meier. And the trick is really to split up Kaplan-Meier into one minus Kaplan-Meier into two additive parts for the respective events. It is very easy. It's a two-line computation really. What you do is you scribble out what one minus Kaplan-Meier is. It's the sum over all event times, and your summand is the Kaplan-Meier estimator previous to the event time in question times number of events divided by the size of the risk set. And that number of events divided by the size of the risk set, you divide that up in number of, let's say, adverse events divided by the size of the risk set plus number of competing events divided by the size of the risk set. And that's all. There's a deep mathematical theory underneath it, which is product integration. So that is fun, but you don't have to do that. And Caspar repeatedly stressed that it has been there in the literature for decades, really. But there has also been a recent literature search published in the Journal of Clinical Epidemiology by Van Wallraven and colleagues, claiming that maybe something, and they looked at the quality medical research papers, right? Not at the tabloids. They looked at the quality medical research papers, New England Journal of Medicine, The Lancet, etc., claiming that maybe almost 50% of all published Kaplan-Meier curves are subject to the kind of bias that we're currently talking about, and with the possible impact that Caspar nicely summarized. So I think, first of all, that is scary too. And that again illustrates that, as we discussed earlier, it's not just about safety. It is possibly an issue in general, but maybe more prevalent with AE data.
Okay. By the way, we'll put all these links to the different things that we discussed about into the show notes. So just head over to theeffectivestatistician.com and then search for this episode and just Savvy, SAVVY, and then you'll easily find this episode. Turning to another point, what is so special about the collaboration here? Because there's a pretty unique setup. So many companies, and also academia, and then all these studies with individual patient level data. How did you manage that?
That's two questions at a time. So I think what's special about it is easier to answer maybe. So for me, but Caspar, please feel free to add. It's a fun collaboration professionally of academia, both academia and companies. And it's really companies, it's plural, it's not just one company, with a joint aim to join forces to solve a common methodological problem. That would be my brief summary. I don't know, Caspar, what would you say?
No, I agree to that. And Alexander, you were mentioning a few points and that speak to the success of Savvy. I think one very important aspect was that a lot of these people, whether they work in academia or pharma industry or even with the regulators, they know each other very well. And there was a certain trust available already from the beginning. And I think that helped a lot. And then there was the common goal. In current corporate speak, you would call it the North Star. You want to make sure that we take steps in a direction where safety analyses are improved. And we have one, I'm thinking of specifically one trial where we observe more AEs, but we also observe a big overall survival hazard ratio. And if you actually account for that, you actually find that the AE risk is lower in the treatment arm. But if you just count the numbers in a very naive way, you find you have actually worse safety signal. And I mean, we want to fix that. We want to give a proper account of treatments. So I think that was another very important point. And then more on the logistical side, I think what contributed a lot to how that Savvy became possible is the fact that we never shared individual patient data. So maybe I can quickly describe the setup. put together data from 17 RCTs.
Right.
And the way this was done was that our academic colleagues, they wrote macros in SAS and R. So maybe start from the beginning. First, we defined a data structure that within the company, you put these trials into that data structure. So which AEs you look at, what are competing events, and then you assign flags and you do that. And based on that data structure, our academic colleagues developed macros, okay, in SAS and R. And these macros were shared with all the sponsors. And then within the sponsor company, you would run these macros, and these macros would then extract these estimated probabilities of AEs for 5 different estimators. And then we would only return these estimated probabilities of AEs back to the academic trial center. And they would then perform a meta-analysis on these estimated probability of AE data. And these then ended up in the analysis I very briefly sketched before, where you then get this underestimation up to a factor of 3, overestimation up to a factor of 5. You look at all these categories. And that facilitated putting together these 17 RCTs. I think we would not be where we are today with Savvy if we would have tried to organize individual patient data sharing and putting it all together in a central place from all these companies. I think that would simply have not worked. And I think this is a learning beyond all the content that we generate in Savvy. This is a template for a collaboration that is potentially also applicable for other type of questions.
Yep. And especially when you started to mention that this is a macro, like the preparation of it. I mean, getting the data and then just playing around, that's comparably easy, but really putting the planning in place and really send over macros that work with, I don't know, a number of, randomized trials. That's just awesome. So it just gives them a different dimension in terms of organization and commitment. So it's a really great story, really.
Yeah. Hopefully, where we in Savvy so far, we have aimed to answer methodological question, but the future plan will also be to address specific therapeutic areas. But Alexander, in terms of management, there are really a number of people that we haven't talked about yet, without whom Savvy would have simply been impossible. It would something that we might be talking about today still over coffee, but would not have happened. And that is, of course, Tim Friede from Göttingen and Claudia Schmoor, with whom, Claudia was a leading force behind that GMDS workshop that I talked about earlier, and that led to that special issue in the Pharmaceutical Statistics Journal. And then almost in parallel, Tim, Claudia, and me, we discussed putting the concerns that were around into an effort like that became Savvy. Well, Kasper and me, we were together today here on the program, but a lot of the hard work, writing the macros, doing all organizational bits, was done by Regina Steke as part of her PhD thesis at the University of Ulm. And so, yeah, Kasper, what would you say? I'd say it's really a big shout out to Regina, Claudia, and Tim. And there have, of course, been people from the industry alongside Kasper who have been very supportive. So initially, when we were kicking around this idea, I had the impression that maybe it's a nice idea, but it doesn't really take off. And then we had an organized session on adverse event analysis at the Central European Network, ISPS, meeting in Vienna in 2017. And then within a month after that, Katrin Kupers of BMS, they committed themselves. And then Kasper was a key player, Roche was a key player, and Valentin Yale from Novartis was a key player. And then things just came together. And as Kasper said, most of us knew each other personally from conferences, workshops. Kasper and me, we actually go back to days in academia in Zurich, almost 15 years, and then it took off.
Awesome. Yeah. It's a great example of collaboration between like-minded statisticians. Yeah, awesome.
Yeah, and it's open source knowledge. Still needs to be organized, but the way we went about it is we came up with a statistical analysis plan, and we turned that into a methodological paper, which has seen the light of day in the Biometrical Journal. Regina Stegherr is the first author. So that is a template for methodological investigation like this that you can transport to other questions, obviously. And it does come with the source code for the in-house analysis, as Kasper explained, that were run at the specific pharmaceutical companies, and it comes with the source code for the meta-analysis that was centrally run at the academic institutions. So the stuff is out there, and it's free to reuse it for related questions.
Yeah, and maybe if I can add to that. So I think we initially, as usual, we completely underestimated what this will mean.
Yes.
Because you would think you define the data structure, everybody puts the trials in that data structure, you run the macros, and 2 weeks later you're done. And then we ended up doing a pilot study. We had a few companies, a handful of companies participating in a pilot, developing the macros, and that took much longer than we thought. If you then run these macros, you just develop them first round, and then you run them, you find this problem. In this trial, we have this very special feature. We need to update that. Everybody's using a different SAS version. Okay, this means that. And so I don't think we want to pretend that this was easy.
No.
But that even makes it a bigger achievement. I think that we persevered and really went through. And a big shout out to everybody involved. So the people Jan mentioned on the academic side, and everybody in the companies who actually put that through and organized the data and applied the macros and fed back what they found. So this is it's a big thing, in my opinion. And maybe just to add one more point, for us in pharmaceutical industry and maybe even in academia, these points that we are underpinning with Savvy, they have been made for decades. A lot of people said it's not accurate what we do. The methods are here. Why are you not using them? So that's one point of view. But if these methods are never used, then at some point, I think you need to try to take a different approach, that people start to use it. And you need to convince everybody involved that what we are doing is not what we are supposed to do. And I think here, that you apply it to real RCT data and that you make it very concrete and you illustrate the impact on labels. Hopefully, sometimes at some point, we will start to see different analysis. And I always use that kind of image of this is safety analysis in regulatory RCTs is like a tanker. And hopefully we can have this tanker turn the direction a little bit. And a lot of people have tried in the last decades, and I think with very little success. And maybe here we can start to change.
I think that goes back to a theme that we have pretty common here in the podcast. It's about you have all the logic on your side, but still things don't move.
Yes.
So you need to have other ways to influence people, and you need to kind of step into their world to show kind of what it means. And I think that is exactly what you did with the actual RCT data to show that, well, it's not about just simulations and made up data. It's about actual compounds, actual, impact in different studies. And it's, showing that really kind of how big the impact can be, threefold, fivefold, and that helps people to understand that, there's really a problem, and it's not just kind of mathematical problems that might occur in some, rare occasions that we don't necessarily need about, and so I think that is especially, what we as statisticians need to become much better at is stepping into the shoes of our audience. What do they care about? And the physicians care about the safety of the patients, and the regulators care about, what they write into their labels. And so showing them, the impact of that in their ways, in their terms, is a really, really powerful way to change the overall game in terms of safety analysis.
Yeah, that's right. I should give credit to Tim Friede, who in 2016 he challenged me. That's all swell, but does it make a difference in practice? And that's something that Savvy has demonstrated on a scale that we have not seen before. I think that is fair to claim. So as Caspar said, it has been there for decades in tons of papers, typically demonstrating matters using simulated data, of course, and then you can come up with anything that pleases you. And again, mentioning COVID-19, there's a paper using simulated data, again, making the very same point for COVID-19 treatments, but right, we're using real data, and that has been there before too, but not on this scale, meta-analyzed across many adverse events and a range of trials. So that is really different from an applied perspective. And probably we're also the first, and then on this scale, as just discussed, also looking at group comparisons, because typically the discussion focus on, let's say, the old Kaplan-Meier estimator, that is overestimating. So if you're overestimating in your experimental arm and in your control arm, what does that mean in terms of group comparison? And that is something that I wouldn't know where to find safe for Savvy.
I think the other point is that you started is, and which is an important thing if you think about change management, is you have, you build not just work with one company or two companies, you worked with many companies. And if you want to change an industry, you need to have some kind of guiding coalition. And I think that is another great thing, that you not stop with, oh, we have Roche on board, that's fine, let's move forward. But you actually moved forward with lots of different companies and were very, very inclusive there. And despite the additional challenges with different SaaS environments and different study setups and additional kind of problems on that side. But now you have, advocates for that in lots of different companies. And that makes things much easier. It actually also makes things easier within the company. Yeah, I know that, when you're just talking within your company and you have this point, it's actually great, but it's even better if you can point to some other competitors and say, they actually do that as well. Yeah. So, the prophet in their own land is usually not heard, but if you can refer to some other prophets as well, it's much more powerful.
Yeah. Yes, of course. And I mean, we want more accurate estimates of probability of adverse events and accounting for all these features. And if we can help establishing that, maybe in 10 years, 20 years, then that would be great. And I mean, Alexander, you mentioned the estimand addendum before. It is possible, although you can argue this is very difficult and you might not be happy with the direction of the estimand addendum, but we see now it has a huge impact, and it is possible to impact things, how we do things, although there is, of course, inertia, because we have all these established ways, established systems people are used to. It takes some effort, but this is for me kind of the illustration or the example that things can be changed, and let's see where we get with Savvy and the analysis of safety data in clinical trials.
Yeah, I think this was the idea why to put together a large group, and personal contacts helped. The typical mindset was that everyone was willing to contribute, precisely for the reasons, Alexander, that you have just mentioned. And I mean, at the end of the day, Caspar, the aim would be to improve guidelines, right?
Where can people actually learn more about Savi? So where can they find out more about the project, about the people?
As innovative as we are also old school, meaning we have no website. I don't even tweet. So you learn about it reading the scholarly papers, and we have been very active at conferences, but there is no website that you can go to.
But there is a nice homepage from Caspar where there's a couple of nice links, and we'll put that into the show notes. Okay, thanks so much. That was an awesome deep dive into Savi, and we had lots of learnings from it. We understood kind of from a methodological point of view, what are the limits of just counting events or looking into the Kaplan-Meier curve. We understood kind of that this is not just an oncological problem in oncology, but it's, widespread across lots of different areas. And that it took a couple of people that had the same goal from both companies and academia to work together, overcome lots of hurdles, move the needle forward. And now we have something in hand that can help us to convince people to apply the methods that, as you stated, have been out there for decades, but now we have really proven how much they can improve things. Any final thoughts from you, Jan?
Well, Alexander, Benjamin, thanks for having us. It was great talking to you. And to everyone who listens to this podcast, I think it's safe to say that we are looking for some volunteers who want to practice with a future aim of looking into different fields of application. So not taking any trial, but let's say look into oncology, specific fields of oncology, and looking into other therapeutic areas to better understand where these methods maybe matter most and what kind of difference we see with respect to subject matter applications.
Caspar, anything from you?
Yeah, maybe to conclude, I think what is always helpful is, and that is the spirit of the estimand addendum, be clear on the objective of what you want to do. Don't just reproduce what you have done in your last trial, in your last analysis. Think about, do you want to do signal detection? Do you want to properly estimate the probability of an AE and then develop the estimand, the estimator, the data collection accordingly? And yeah, use the methods that we put forward in Savi and use to learn.
Awesome. Thanks so much. This show was created in association with PSI. Thanks to Rain, who helps with the show in the background, and thank you for listening. Head over to theeffectivestatistician.com to find the show notes with all the different links. And you can also learn much more through this podcast and through other material on the homepage to boost your career as a statistician in the health sector. Reach your potential, lead great science, serve patients. Just be an effective statistician.